Hey guys, this is my dissection of a sheep eyeball video. So to start out on the external features, we have the anterior and the posterior side. On the anterior side, we have this cloudy grayish structure. That is the cornea. This is where the light enters through the external world. And then if I move this out of the way, we have the sclera. That's that white ring that was around the cornea. The sclera is more of the protector of the eyeball. It helps to protect the inner structure and helps maintain the eyeball's rigidity. And then on the posterior side here, on the external features, we have this tail-like structure. This is the optic nerve. This is where the nerve signals go from the eye to the brain to relay sensory information and neural impulses. So back to the anterior aspect here. When I cut this in half, a bunch of fluid came out. That's the aqueous humor. This is the area that fills between the lens and the cornea of the eye, and it helps to maintain the shape of the water. But what also came out was this gelatinous structure and this spherical structure. This spherical structure is called the lens. The lens is where the light gets focused as it comes through the cornea and the pupil. And then this gelatinous structure right here is the vitreous humor. This is more of just viscous fluid that helps to maintain the shape of the eyeball. This is what the lens sits on, and the lens sits in this indent on the vitreous humor called the hyaloid fossa. It just helps to support the lens a little bit better. This black ring here is the vitreous body. That's what the vitreous humor sits against. It's a cavity between the retina and the back of the lens. And then if we continue on, there is a hole in here. You can see it there. This is actually the pupil. This is where the light enters through the eye through this opening. On both sides, if the lens was sitting in here, we'd have ligaments that sit here. They're called the zonula solaris. They suspend the lens and stretch it to focus the vision. So those would be right in here, as well as the iris. The iris is the diaphragm that helps to regulate the size of the pupil, so it'll either shrink it or increase the size. Moving on, if we go to the posterior side again, we have this, there's a white film here. It's more of a sheet. I have it crumpled up into a ball so I could see other structures. But that's called the retina, and the retina is the light-sensitive portion of the eye that is where the rods and the cones sit. The rods are the receptor cells that are responsible for perceiving differences in light intensity, and then the cones that sit in here are the receptor cells that are responsible for perceiving color. So underneath the retina, we have this tapetum lucidum. That's this shiny iridescent portion of the tissue. This helps with night vision. So we as humans do not have night vision, but this is for animals. So that helps them be able to see when it's dark out. And then we have the choroid, which is a thin sheet that's held between the retina and the sclera. So that's like this outer portion underneath this white. That's all it is. It's just a thin sheet. I have it pulled back here so you can see it a little bit better. Right there. And then last but not least, we have the blind spot. So the blind spot is where no light reaches. The rods and cones aren't there, so can't detect any light. That's actually underneath this, right where the, right before the optic nerve. And so that's just where the area of the retina is that the receptors can't conform and they can't perceive that there's any light going on. So yeah, that is my dissection of an eyeball.